Okay, hello folks, good afternoon, and thanks for joining us for another TESS seminar here. Before we get into the introduction for our speaker, I'd like to just make a couple of quick announcements. The TESS retreat, you're going to hear a lot about this in the coming months and weeks, and you might as well just sign up for it. That way we'll stop harassing you, coming to your house in the middle of the night. Why haven't you signed up yet? So, and Yoko asked me to mention a couple of things here. Firstly, that next week is a little bit unusual in that we're going to have two seminars. So one of the seminars will be at our usual time, Wednesday at 4 o'clock. And this is a talk by Kieran Sims, who's one of our faculty here doing a lot of interesting work on environment, society, that type of thing. And looking at BRI, which I guess I'm assuming is the Belt and Road Initiative, as cognitive empire, epistemic violence, and ethno-nationalism in northern Laos. Okay, so you want to come just to see what that title actually means. And then the next talk that we'll have will be Thursday at 4 o'clock. Thursday, not Wednesday. And this is this is from Lisa Davenport, who's of course John Terborg's partner. Now, John Terborg, of course, is that eminent Princeton and Duke University scientist, MacArthur Genius Award winner, who is affiliated with JCU and who was coming out here every year with Lisa. And now, because of the COVID thing, they haven't been here for 2 or 3 years. But anyway, they're back and they're keen to engage with us. So John Terborg, famous guy, and Lisa Davenport, also a very respected scientist. Anyway, she'll be talking about her work on the migration of the black skimmers. So she's doing a lot of cutting edge stuff using radio telemetry. Okay, now I have to get out of this. Yoko, let's see here. Okay, can you help me, please? This is not, can't get that to, oh, there we go. There, oh, no. Is that it? Okay, that's it, great, that's it. Fantastic. Okay, well, it's a real pleasure to introduce Jamie Cleverly. Jamie's going to talk to us about some of her work on tropical rangeland, the tropical rangeland supersites. SuperSite, as you probably know, a lot of many research sites and research infrastructure in Australia are associated into various kinds of groups or organizations, overarching organizations that are sort of trying to provide scientific coherence and consistency. I hope I got that about right. But anyway, Jamie's going to talk to us about that work. Jamie's from the US originally. She got her PhD at the University of Nevada, Las Vegas, which, by the way, I will mention is the archenemy of the far superior Boise State Broncos in football. But anyway, the fact that our universities don't like each other, that's not really here nor there. Anyway, very happy to turn things over to Jamie to talk to us about JCU's Fletcherview Tropical Rangeland SuperSite and more about that. Thanks, Jamie. Yeah. Okay.
Do we need to drag it over?
Yeah, I think so.
There we go. I'll just have to look at it from here. So today I want to talk to you about what we've been doing here at JCU. And this has been a fairly intense project involving quite a lot of people, including Lucas here. And what we've been doing is we've been setting up the Fletcherview Tropical Rangeland SuperSite on behalf of TERN and the Queensland government. And so I'm going to be talking about what environmental research infrastructure we've been developing here at Fletcherview. And I'll also mention that my lab here is called the Center for Plants, Atmosphere, and Water, in case that is of interest to anyone. I'm affiliated not only with JCU and TERN and Queensland government. Of course, TERN is ultimately funded by the National Collaborative Research Infrastructure Strategy of the Commonwealth government, and also OzFlux. And so I'm going to be talking about all of these things at one point or another. I'm going to start with TERN though, seeing as how they are sponsoring, kind of sponsoring the show. TERN is Australia's land observatory. And so a lot of the environmental infrastructure related to ecosystems in Australia is being put in the field and provided by the Terrestrial Ecosystem Research Network. TERN operates at three levels. It operates at local scale monitoring. So these are the super sites. These are the sites that try to get at why and how ecosystems are changing. So really focused on the ecosystem processes or the ecological processes that drive the patterns we see in ecological systems. Then at the intermediate scale, we have the regional scale monitoring. These are largely a network of plots, hundreds and hundreds of plots, some of which are also at the super sites. And so the super sites in that local scale monitoring kind of brings it all together into one place. These plots really focus a lot on biodiversity and soils. And so they go out there every 10 years, they set up a plot, then in another 10 years, they'll go back and revisit it to look at some of the long-term change that you might be seeing in the landscape and in the vegetation. So they are really focused on what is changing and how much it's changing over longer time scales. And then the largest scale monitoring that's being done by TERN is the continental scale monitoring. So this is more like satellite remote sensing, gridded soil products, something that covers the entire continent in a fairly continuous, even fashion. And so all of these come together to provide a picture, both a picture and data of what is going on in Australia with respect to its ecosystems and how they're changing. The other key facet of this infrastructure is that all of these data are publicly available. So anyone can use these data to enrich their own research. So you'll see today that we might have a lot of contextual data that you might be able to use in, say, your faunal studies, because we're measuring temperature and we're measuring humidity or whatever else might drive population changes in what your organism might be. It's as I was saying, it's a distributed environmental research infrastructure. And you can see those hundreds of plots that I was talking about in the little green circles. The super sites, we have about 16 of them now. They're distributed across the country and pretty much all of them have flux towers, which I'm going to be getting into, but not quite yet. And then a lot of the continental scale monitoring CalVal sites are co-located with these plots and with these super sites. Quite a bit higher density in southeastern Australia. That's where the people are, and so it just kind of happens that way. So for a more specific view of what TERN is and what TERN provides, it provides flux towers. It provides, so we're using sensors, surveys, and tools to generate this picture of what's going on in the landscape. And so we have flux towers, heat flux plates, radiometers, anemometers, infrared gas analyzers. Now, this probably doesn't mean a lot to any of you. What we should really be saying is we're measuring CO2 density, we're measuring water vapor density, we're measuring the exchange of carbon and water and energy between the ecosystem and the atmosphere. That's what those first set of sensors are. Then we have things like Cosmos soil moisture meters. So these are really good for satellite remote CalVal of satellite remote sensing, because it's really hard to get a good handle on soil moisture because of its variability in space and time. And you can't measure it everywhere in Australia. It's a vast continent. We have some groundwater bores, not at every site, but at some sites where they're appropriate. And we'll be talking about those in a little bit. Phenocams are really becoming to be a crucial tool in our toolbox for understanding how ecosystems are changing, because they can actually tell us when the vegetation is green, when it's reproductive, when it goes brown. And then we can start looking at some of those ancillary data and say, well, it went brown because of this event. And so then we can start looking to the future to try and predict how vegetation across Australia will respond to things like droughts and heat waves and so on. We also have from the plots, we have over 100,000 cataloged plant and soil samples that are available for analysis by anyone who wants to do it. You probably would have to travel down to Adelaide, so you might not want to do that in the winter. But otherwise, there are really quite a lot of opportunities. For example, with the soil samples, opportunities for looking at eDNA and finding novel drugs, which these samples have been used for. We provide standardized monitoring methods, and those are actually currently coming together into a single one methods compendium, so that basically everyone can be on the same page when it comes to looking at these kinds of things. And so on and so forth, including the data products. I'm not going to really spend too much time on them. Probably go pretty quick here. So our TERN's data are used by a vast number of different kinds of people. The most important are researchers at universities, or the most numerous, I guess you might say, but also researchers from international researchers from other institutions. Government departments are really important because that's one of the key factors for NCRIS, for the federal funding. We have to contribute back to the Commonwealth, and we do that through government departments. But also there's some toeholds in industry and non-commercial organizations. We are also working towards the global sustainability goals, the global sustainability goals. The one that figures in largest is number 15, life on land. And so we're trying to provide data which can contribute to improving our sustainability for life on land, for can't read some of those others from here, so I will just let you look at that, and then I will move on. Finally, for further information on TERN, we do have a publication, came out in 2019, that is specifically about TERN, but also addresses the global challenge of forecasting ecosystem responses to climate variability and change. And as I go further into this talk today, I will talk a little bit about how Australia fits into understanding climate variability and change, as well as how Fletcherview is expected to add its piece into that puzzle. So we have 16 ecosystem processes SuperSites. I've marked a couple here. First is the Alice SuperSite. That is one that I established in 2010, so 12 years ago. And 12 years ago in just a few days, in fact. And but I'm not going to be talking about that one so much today because our newest member of the flock, which gives it the largest symbol on the map, and I don't have any arguments with that, is our Fletcherview site. So now I'm not going to spend too much time on the super sites in general. I'm going to jump right into the Fletcherview Tropical Rangeland Super Site. This is funded through a co-investment scheme between NCRIS, TURN, and the Queensland government. And so both NCRIS and Queensland government put in money to, amongst other things, to set up the Fletcherview Tropical Rangeland Super Site. As you probably all know, the Fletcherview field station is on a cattle station. It's a working cattle station that's used a lot by the veterinary students down in Townsville. But we are really keen in the larger administration at JCU to expand our research presence at the field facilities. And that includes Fletcherview, and that includes the DRO and a couple others. So the first thing that was done at Fletcherview was to put in biodiversity plots. These are those plots in the, and we call them surveillance plots. These are the plots at that middle scale of observation that are focused on biodiversity. And we have 3 at the Fletcherview site. We have two down by where our core operations are, and then a third was put in the red basalt community, which is really quite a different vegetation type. So for completeness, we have all 3. I'm mostly going to be focusing on QDAEIU 0008, which is what we're going to call our core plot for the super site. So this is where we're really going to be intensively measuring things. The other thing that I wanted to note here is on the map on the right, all those little yellow dots up near Cairns at the top, and then Fletcherview just north of Charters Towers. Those are all of the surveillance plots that we have in this area presently. So and that includes up towards the tropics and the tablelands. So Fletcherview is also located near the Burgeoning River, which makes it a highly significant site in the paddock to reef program. And we're working with some of Queensland government who are involved in that as well. So looking a bit more closely at these biodiversity plots, here you can see the 3 plots, and what they're set up as a 1 hectare plot. So 100 meters by 100 meters. And that's pretty much what you can see from this figure. Notice that I've also labeled the mobile tower. And I'm going to be getting into that shortly. But pretty much all of the maps should reference to the location of that mobile tower, just so that you can see where you are. It's a subhumid tropical savanna, and this here, what you're looking at here, is the core plot. So discontinuous tree cover with lots of grass when it's wet enough, and then we also have a bunch of shrubs. And in fact, the shrubs are dominant in this plot. So we have the Carissa shrubs, which are native, so we can't just remove them. but they form the largest portion of the vegetation in this plot. And then we have a whole heap of tussock grasses, forming that grass component of the savanna. And our main tree is the Eucalyptus brownii, or Brown's box. So we're actually going to be focusing. We're largely going to be focusing on the Brown's box and the currant bush, the Carissa. Sorry, no. We're focusing on the Brown's box and the tussock grasses because, and I'll show you why in a little bit, but the answer is actually because we're interested in cattle forage as well. So we're not just doing ecology, we are also interested in the agroecosystem in general. We're not only using these plots to collect biodiversity, plant biodiversity data. We're also working with Will Edwards, who has put in these little pitfall traps to collect ants. So we're looking at ant biodiversity. The SuperSites also have A2O acoustic monitoring set up. So for people who are interested in birds and other things like that. And as well, they recently put in the Anabat detection system for detecting bats. So if you're interested in biodiversity of bats, this is also a good place to go. The key to all of this, because I'm obviously not a biodiversity specialist, I learned about it when I was at university, but hey, I've moved to other places. But others are welcome to integrate with our contextual data and use this site as a springboard for doing your own biodiversity work. So the next thing that we're doing is pasture biomass monitoring. And we've set up, as you can see with the four yellow pins to the north of the mobile tower, we've set up an exclosure plot so that we can monitor the effect of grazing and the biomass production in the absence of grazing. So this is something that's really important for some of our rangeland modeling, which is one of the reasons why this is such an attractive site, because we can integrate across different fields in this way. And so what you can see is the exclusion plot when they're putting it in after we'd had a fairly long dry season, and there's really no grass biomass to speak of. So it's really a good time to put in an exclosure plot. And then you can see in the middle photo where they have put in the fence, the exclosure fence, and you can see what's happened with the grasses after the rains came. Sometimes it's even easier to see these things if you have a graph. And so what you're seeing here is just before December of 2021, when that exclosure plot was put in, there was pretty much no grass at the site because that's what it's like during the dry period. But then you can follow it through, and these are in the exclosure plots, so these are not being grazed. And so it was sometime in early to mid-January, or maybe even late January, when it really started to rain. We got kind of a late wet season this year. And that's when the pasture biomass just skyrocketed. But then something really weird happened, and that's that it kept going up in April and May, because like with cans, the Fletcherview field station has received in the last year a dry wet season, but an unusually wet dry season. And so the rain just came back, and the pastures loved it. We're also interested in the soils. The soils are key for understanding the pasture pasture biomass and growth, because they're key to understanding how we can translate from precipitation to soil moisture to growth. We have the soil types are very well characterized at this site, at least at the surface. It's dominated by clays, a lot of cracking clays. There are some sandy clay loams, but for the most part, the site is dominated by clays. We're not stopping there though. We've been working with Queensland government they brought out their drill rig to take soil cores down to 1 meter depth at a variety of locations, starting from near the mobile tower and then going north from there. We're still waiting for the data to come back on these, but we're really quite excited to be able to characterize the site in a lot more detail than it's ever been characterized before, especially at depth. It's not a lot of depth, but it is depth. Now, I'm going on a tangent here because this is actually nocturne. When I was at UTS, we got funded on an ARC LEAF proposal to build an Australian Critical Zone Observatory network. And so that's why you see Alice Mulga in the little black and white circle there, because, but that has now moved to Fletcherview. So Fletcherview will be the Critical Zone Observatory, not Alice Mulga. So it came with me. Essentially, the critical zone has been well studied in the USA and Europe. They've had some very successful programs to study the critical zone. So-called because it's that slice of the biosphere that makes life possible. So essentially from the groundwater and bedrock up to the tops of the trees. And it's the interface between geology, hydrology, soil science, and ecology. So it's really a multidisciplinary view of why things work the way they work in an ecosystem, and both the above ground and subsurface portions of that ecosystem. We do have partnerships, so it's not funded by NCRIS programs, but we do have partnerships with TURN, with Oscope. Oscope is the NCRIS facility that looks at the geology. So we're kind of stuck in the critical zone. We're kind of stuck in between those two end members. And that's what we're integrating across TURN and Oscope. And also the former groundwater facility, but they're no longer with NCRIS. This critical zone infrastructure that we're putting in is really focusing on the lithology, but also the groundwater conditions. So we're really hoping that we find groundwater at Fletcherview. We don't know if there's any under there yet. But if we do, we have a heap of sensors to provide data to anyone who's interested on the groundwater conditions. But what's most exciting is this vadose zone monitoring system. And these have been rolled out in a few places around the world. And they come out of Israel. So they're going to be traveling to Israel when we have the drillers finally all lined up. And what they do is they put in a sleeve down this bore. It's about a 35-degree angle bore. And in that, they have soil moisture sensors. They have sampling ports for gas and solute sampling. And so almost anything, if you are a vadose zone hydrologist or soil scientist, probably almost anything that you might be interested in, we can accommodate you with either samples or data down these vadose zone monitoring, down this vadose zone monitoring system. They just call it VMS for short. We currently, right now, have the permanent ERT array in the post to come to Fletcherview. So probably in the next couple or few weeks, we will be putting in the permanent ERT array so that we can finally get a picture of what's going on underground to as much as 70 meters, I think is what they got to in South Australia. And so that should be able to tell us if we have groundwater, how deep we need to go to get groundwater in our vertical wells. And then that comes with its own communications. So everything that's measured will come back to us and be freely available. Samples you have to go out and collect yourself, just as we do. So now we're actually looking at two images. It doesn't look like it, but we're looking at two images. One on the left is the wide area view. So you can see the Fletcherview research station and the mobile tower with respect to where that is. But then in the smaller illustration on the right, you see A, B, and C, and those are 3 dots with a line. That's approximately where we're planning on putting our bores. At least that's what we told the drillers, and might as well just go with it if that's what you told the drillers. The line shows you the direction which that angle bore will be going down. The brilliant thing about that angle bore is that if we can confirm that we've gotten below the root zone, then those soil moisture sensors can actually start to tell us something about drainage, which is something that's really hard to work out because you have to be able to measure soil moisture below the root zone. Otherwise, you can't tell how much of the water that's there is being pulled up by the trees or continuing down towards the water table. So this is really exciting if you're a hydrologist. I can promise you that. So yeah, now I got to the point where most of you who know me, know my background will say, but aren't you a flux person? Don't you do fluxes? Well, yes, I do. And so now we're going to talk a bit about the core bit of the infrastructure that has fascinated me for the last 25 years. Now, I know that the photo on the left does not show anything to do with fluxes. But what I wanted you to see was the woman on the right, that's Jo Owens. She's I'm one of her PhD supervisors, and she's been doing a lot of the work to analyzing a lot of the data, which I'm going to show you from this flux tower today. So couldn't get most of this done without Jo, and so I wanted to acknowledge that right up front. So what you see here on the right is Mirko Karin up on top, lifting the sections of the tower up. So it's a pneumatic system, but it takes a lot of strength to get those sections up in place. And Mirko's been a champ at that. And so that's him up there, and I'm down on the lower platform, keeping the cables that need to rise up with the tower straight, so that we don't have any of the problems which we've had recently with broken cables. But you know, what are you going to do? So fully retracted. So this is before we put the flux gear up to 25 meters, mostly so that you can see the instrumentation on the top, because that instrumentation on the top is really what it's all about. And when I say what it's all about, we're measuring the fluxes of carbon, water, and energy. What that means in just regular terms is that we're measuring kind of the balance between photosynthesis and respiration. Then we can use some models to try and disentangle them. We're measuring evaporation, and we're measuring evaporation whether it comes from the soil or whether it comes from inside a plant, inside a leaf. And evaporation, which occurs within a leaf, of course, is called transpiration. We're also measuring a full energy budget, including radiation, so solar radiation, thermal radiation, convection, so that's the the turbulent transfer of water vapor and heat, and then conduction of heat into and out of the ground. So all of these components is what makes up a flux station. And they're usually put on towers because we need to measure everything well above the canopy height, because what we're measuring is the flux of these things. So the photosynthetic flux from the ecosystem to the atmosphere, we have to be above the ecosystem before we can make that measurement. So the mobile tower, we have a really nice power hub that was built for us internally here at JCU, but there are a heap of ancillary measurements that we need to make to not only, but to most importantly, to understand the fluxes, but also to start to get a handle on what's driving changes in the ecosystem processes and ecosystem patterns. So that includes things like full meteorological conditions. So on the mobile tower, in addition to the flux sensors, we have various meteorological sensors like temperature, relative humidity, wind speed, and so on. But in addition, we need to measure how much rain is falling because that's a very large component of the water budget. We have phenocam set up, as well as several soil sensor arrays. These soil sensor arrays contain a heat flux plate so that we can measure the conduction of heat through the soil. They contain a soil water content reflectometer so that we can measure the soil moisture. And they contain soil temperature probes so that we can very accurately track the temperature of the soil. And pretty much all of these soil measurements are crucially valuable for the station manager. This is what they need to know to be able to predict what the forage is going to be and then move their cattle around appropriately. So we actually have some data for this talk. Now, there is a extremely good relationship between precipitation and soil moisture. You would expect this, and it's not quite the same during the wet season as it is during the dry season. You kind of expect that too. But pretty much whenever it rains, you get a spike of soil moisture right near the surface. And then as you have evaporation and you have transpiration occurring, you quickly, following an exponential function, the soil dries out until the next rainfall. Now, this is important for what's coming because we're going to start talking about disentangling what's driving changes in evapotranspiration, or the combined evaporation and transpiration terms. So the first hypothesis is that it's soil moisture which drives variability in evapotranspiration. The second hypothesis is that it's rainfall. So you can see that in general, evapotranspiration does follow the pattern of precipitation, but the magnitude of this response is different during the wet season than it is during the dry season. So especially looking at that big precipitation spike in early May, there wasn't an equivalent spike in evapotranspiration following that. There was a bit of a one, but not as big of a one, because evapotranspiration really relies on temperature and the vapor pressure deficit in the air. And during the dry season, those are always lower than during the wet season. And so there could be a lot of things going on. This is a site that actually gets more rainfall than it can use for evapotranspiration. So we have to figure out where that water is going. Is it just sitting in the soil until the plants can transpire it? Is it infiltrating deeper? We don't know yet, but we hope to at some point. So another spanner in the works is that it could be the response of the vegetation that drives these patterns in evapotranspiration. And there is a really good relationship that you can see here albeit kind of lagged between what we call the green chromatic coordinates. So basically how green the grasses are, because this camera is focused on the grasses, the grass layer. So evapotranspiration comes up a little before the grass grows. So here we're probably talking about the possibility of separating out soil evaporation from transpiration in these grass plots. At least crudely, I don't know yet, but we'll see. And maybe we have some models that can help us. So essentially, we now have three factors that we know are related to how much water is evaporated from this site. We have soil moisture, or the availability of water to evaporate. We have precipitation, and we have the vegetation itself. And they probably all interact to make it especially complicated for us. What's important about this is that, so this is just a burgeoning site. We're just getting started, but we're part of a global network called FluxNet. And this global network has hundreds of towers all around the continent. And these data are also available for free, with the caveat that they're cut off in 2015. So they haven't prepared the next version yet, but we're expecting that at some point they're going to be a bit more timely and produce the data, say monthly or something like that, which would be brilliant. FluxNet is actually a network of networks. Some of those networks are OzFlux, Ameriflux, AsiaFlux, ICOS. There are a few other smaller ones. I don't remember what the one in Africa is called. And then AsiaFlux is also a network of networks because it includes JapanFlux, CoFlux, and ChinaFlux. But for repositories of data which are related to ecosystem processes, this is the place to go. This is the place to see. What it does is it makes possible a better understanding of ecosystem processes at a global scale. So this is a paper that we published last year in Nature, which was really exciting, looking at how various factors combine and contribute to controlling the fluxes that we measure. And so the highest amount of variability is related to leaf area index, so how much leaf biomass is produced, or leaf area is produced over a site, the above ground biomass at a site, and the height of the canopy. And then as you can see, these factors build into relationships at differing levels of variability. So sometimes the lower levels of variability actually explain most of the relationship. And so it's really exciting, but there's still a lot more that can be done with these large global data sets to which Fletcherview will be contributing. I mentioned OzFlux because we are also a member of OzFlux. OzFlux is the Australia-New Zealand Flux Monitoring and Research Network. And just earlier this year, we published our 20-year anniversary paper talking about the lessons we've learned 20 years of making these flux measurements. Can't wait to get Fletcherview on the map. So didn't have enough space on here for all of the authors. So I hope they'll forgive me that I only put 1.5 lines up. Seemed to be the most important to me because my name is in the top line. But some of the lessons learned relate to extending our understanding of the climate space in Australian and New Zealand ecosystems, ecosystem recovery from disturbance, drought and heat stress, resilience to climate variability, climate impacts on agroecosystems, synergies with remote sensing, synergies with modeling, and long-term measurements to detect decadal and climate change effects. So we know that Australia is arid and hot. And one of the ways that we know that is, I mentioned before, the vapor pressure deficit. In most parts of the world, you say your vapor pressure deficit is 5. They'll say, oh, wow, that's hot and dry. We get up to 7 or 8. And so it's like a whole new ball field, whole new cricket pitch, really, here in Australia. And that has a large effect on the functioning of these ecosystems, specifically stomatal conductance, which is what we showed in this paper. But more generally, looking at that inset figure, the gray shows you the climate variability for the whole globe. And then the blue shows you the climate variability for South Australia, and the red shows you the climate variability for North Australia. So it doesn't matter where you are in Australia. Australia has a more variable climate, more extreme climate than pretty much anywhere else in the world. And so this allows us to answer questions about climate change that are just starting to emerge on other continents. And that's really significant, I think. It's possible that we may deal with a tropical cyclone at Fletcherview. So these kind of data do contribute to understanding how ecosystems respond to disturbances like tropical cyclones. They also, there's also the possibility that we get bushfire out there. I know that it'll be fought because of the cattle and all, but it's still a possibility. And so our data could contribute to a better understanding of fire ecology in Australia. Also, we've used a lot of sites to look at drought and heat wave, and we are planning drought experiments with Joe at Fletcherview, where we found that there's actually kind of a ameliorating effect of the wet eucalypt forests in southeastern Australia relative to the drier forest. So the drier forest, they get a heat wave and drought, they kind of shut down and start to overheat, whereas there's enough soil moisture in the wet eucalypt forest that it can continue transpiring and actually cool the area and ameliorate that heat wave that's occurring. Some work that we did on agroecosystems, so it would be really great if we could start adding Fletcherview to the mix. And yeah, mostly looking at predictability of the fluxes here. And we're also interested in remote sensing, especially remote sensing of photosynthesis, as well as phenology. And we participate in calibration and validation of NASA and ESA products. Finally, modeling. Modeling is a major focus for Jo, and she's been doing modeling for quite a while, particularly pasture modeling. So we're looking with Fletcherview to expand our scope in that area too. And I kind of rushed through those because it's getting a bit late, and I thought everyone would like a beer or a snack. So thank you for your attention. And if there are any references in there that you'd like, shoot me an email and I'll pass them along.